overview of certain aspects of various techniques and how these techniques are going to help in a protein analysis. Okay. So the session has been divided into 3 sections. The first, I will be covering the basics of what is a protein and why protein analysis is very important for in a field of medical, food, or we can say in the pharmaceutical area. Next, what are the different protein analysis techniques? Just introductory, and the details of all these techniques will be offered in the course. Okay, and the last I will be discussing the scope and opportunities if you are interested in moving further in this particular area of protein analysis. Okay. So you all know that all living organisms have a central dogma. Okay, central dogma. I think most of you will be familiar with the term central dogma. Central dogma is a flow of information. Information which flows from a DNA to RNA through a transcription, and then from RNA to protein through a translation. Okay, so the protein is a very important part of a body system because of it, the biological role or the biological functionality which it provides to the body. Okay. Now, this protein is the most abundant in the cells, and they are highly specific to a particular organism. Okay. So if I ask you that what is a protein, how can you define this protein? Can you tell me what is a protein? What is a protein? In a day-to-day life, you all come across this term protein. Okay. So what is a protein? Okay. Chain of amino acids, right? Building block of amino acid. No, building block of amino acid. No. Polymer of peptides, right? Building block of human body. Say someone has written building block of amino acid, polymers of amino acid, right? Bonded with peptide bond, right? Okay. Chain of amino acid, Shruti, made up of amino acids and peptide chain. Polymers of amino acids. Okay. So many of you have mentioned that that it is a polymeric unit. Okay. So, and the basic monomeric unit of this polymer is the amino acids. Okay. Now, if I combine all those things which you have mentioned, I can say that proteins are the abundant components in a cell, except storage proteins. You know, there are several storage proteins in our in the living cells. So apart from that, all other proteins play a very important role in the biological functions, like they play a very important role in biochemical pathways by acting as enzymes. They act as a transcription factors, they act as a receptors, they are antibodies. So there are different type of roles which are played by a protein. Okay. And if we look at the elemental part, the elements which are present in this protein, if I say it is composed of hydrogen, carbon, nitrogen, oxygen, and sulfur. Okay. So hydrogen, carbon, nitrogen, oxygen, you all know these are the basic components, elements of the amino acids. You know, amino acids are mainly composed of amine groups and the carboxylic acid, carbolic group, and there is a R group through which there can be any other group can be attached. So overall, there are 20 alpha amino acids are there. Some of you have written building block of amino acid. That was not a correct statement. The correct statement is that these amino acids are the building blocks of proteins, means amino acid attached through the peptide bond, okay, and through this peptide bond, there will be number of peptide bonds linkages will be occurring, and at the last we'll be getting a polypeptide chain, and that polypeptide chain can be termed as a protein. Okay, so those all of you which have mentioned that it was a building block of amino acid, that is not a right statement. Amino acids are the building blocks of proteins. Okay, now if we look at this proteins, all proteins differ in their type, number, and sequence of amino acids. You know, amino acids are different types. They are polar, they are non-polar. These 20 amino acids can be differentiated into polar, non-polar, acidic, and basic amino acids. Okay, so and because of their R group, you know, they have a R group, and on the basis of this R group, their molecular mass varies. Okay, so the difference in the amino acid residue is going to define the molecular mass of a protein. Apart from that, apart from this changes related to your molecular mass, this amino acid also governs the structure. Okay, so what type of structure a protein is going to take is highly dependent upon the amino acid sequence which it has. Similarly, the biological functions. Okay, so different biological functions which a protein possess is because of its amino acidic sequence. Okay, so and apart from that, there's certain physicochemical properties associated with different proteins that is also related with amino acids. Okay, so the three important aspects which we have to note from this slide is that we have to focus on the elements. Okay, we have to focus on the amino acids, and we have to focus on the peptide bonds, because these three elements, amino acids and the peptide bonds, are going to play a very important role in the analysis. Okay, is it clear? The elements, we can analyze the protein in terms of the elements which are present. We can analyze the protein in the terms of amino acids which are present, or we can use this peptide bond for cleavage to analyze the protein. Okay, so these elements, amino acids and peptide bonds, are very important when we are working in the terms of protein analysis. Okay, protein analysis, if I define, is you can see it is a broad range of, or you can say a use of experimental methods or techniques that help us in detection. Okay, detection means if in a sample our protein is present or not. For example, if you take a sample from a microbial source, you take a microbial cell lysate, or you take a extract from a plant, or you take a food sample, any food sample, or you take your blood samples, or tissue samples, animal tissue samples, or a urine samples, the first step is that we should know that is there any protein present or not. That is called detection. That is detection of a protein. Okay. After you detect that there is a protein, next steps comes the isolation of protein and its purification. The isolation and purification of protein is highly dependent on the targeted protein. Means first you have to target your protein. Your objective is to fix a protein, then only you can go for isolation and purification. Okay. After the isolation and purification has been done, you need to identify the protein. Okay. Identification goes simultaneously. It can go simultaneously with isolation and purification also. And after the identifications, next comes the characterization. Characterization is, you can say, a very important in terms of when we want to know the protein structure and function. Okay. See, there are number of proteins, and every protein will be having a different amino acid sequences. So there can't be a universal work methodology. So for every analysis, every type of protein analysis, we have to define a specific set of conditions. Okay, like if you're working in the microbial cell lysate, the work methodology will not be favorable for in the case for animal samples. Similarly, if you're working with a plant sample, the conditions, the working methodology will be totally different from that of microbial. So for every type of sample which you are taking, the protein analysis method should be performed in a defined set of conditions. Okay. And next question comes, why we should go for this protein analysis? Okay. See, protein analysis is a very crucial to understand, or we can say elucidate the function of proteins. Okay. And understand the impact of their presence or their absence or their alterations. Okay. Let me give you a very simple example of COVID-19. You know, there are a number of variants of this virus. You know, how does this variants are defined? Can any one of you tell me what are the what is the basis of defining these variants of this COVID? Right. Many of you mentioned about spike protein. Okay. So from here, you can say that unless and until we have a knowledge about spike protein, we cannot define the variants of COVID-19, right? So how does we can define, how does we can say that this is our protein and these are the changes which has occurred in this amino acids? You know, someone of you has mentioned the mutation, right? Mutation is also because if the mutation is occurring in the genetic sequence, okay, I have talked about central dogma, right? So if mutation is happening in the genetic sequence of a virus, what will happen? The DNA will transfer this information to RNA. The changes, alteration has taken place. And through this alteration, the protein will be synthesized with different amino acid residue. Okay. So for that, see, Abhyakt Guru Dash has, I'm talking about that. I'm talking about central dogma. Okay. So if you have a knowledge about central dogma, then only how does spike protein get these changes? The spike protein will be getting these changes only because when there will be change in the nucleotide sequence. If the DNA sequence, if the genetic sequence is getting changed, then only the protein will be synthesized, which will be having an altered amino acid sequence. Okay. So first of all, we should, by protein analysis, we should have a proper knowledge about our amino acid compositions. Okay. So why crucial? Because if we are knowing the amino acid composition of our protein, then only, or sequence of our amino acid protein, then only we can say that there has been certain changes or alteration in the, you can say a mutated form or a mutated, our mutation has taken place into the virus. Okay, this is just an example. Now, similarly, if you remember this Omicron virus, they were telling that this protein, the spike protein, has a high efficiency of binding to the receptor. Okay. Do you have this knowledge that Omicron has been told that this variant spike protein have a high efficiency of binding? Okay. Now you correlate this efficiency, high efficiency of binding, with the change in the amino acid composition and the functional property and the biological activity. Means if you are analyzing any protein amino acid sequence, then you can correlate these changes with the difference in their biological activity as well as the functional property which they are going to deliver into the body system. Okay, so that's why protein analysis plays a very important role, not only in the terms of, you can say, in the in the case of COVID-19. But imagine antibodies which are you can say produced, how they are produced. Unless and until we are able to sequence the antigen, unless and until we know the process, proper amino acid sequence of an antigen, we will not be able to design an antibody. Okay, so protein analysis is a basic step, okay, in the field of molecular biology, or we can say in the field of proteomics. Okay, now apart from this, is it clear to you what I have told, how amino acid composition is going to affect your biological activity and functional property? Now, after you have known the amino acid composition, next step is the protein structure determination. You all know that the protein have 4 structures. The first initial structure is the primary structure, which is a single polypeptide. Next is your secondary structure, then tertiary, and then quaternary. Okay, and this structure provides a stability to a protein. Okay, so you all know about insulin. Okay, another example I'm giving you, insulin. You all know about insulin. Insulin is produced by a recombinant DNA technology. Okay, and it has a 2 chain, A chain and B chain. Okay, and these A and B chain are attached together. Okay, so when it was produced in a bacteria, I'm talking about E. coli, when the insulin was expressed in E. coli. In one E. coli, they express the A chain, and in the another E. coli, they express the B chain, and then they purified it. Okay. After purification, what they do, they chemically join these 2, A chain and B chain. Okay. Unless and until, I'm telling you another, unless and until you will not be able to know the insulin, okay, structure, you will not be able to go for its expression, proper expression. Okay, so for proper expression of any protein molecule in a microbial system, or in an animal cell culture system, or in a plant cell system, we need to have a proper understanding of its protein structure. Then only your process of recombinant DNA technology or expression level, or you can say its utilization, will be effective. Next is the protein mapping. Mapping is the, I can say, as a way of studying the expression level of protein in a specific cells, or you can say tissues and organs. And this help us, this protein mapping can help, normally it helps us in understanding the, you can say, various stages of any disease. Okay, not only disease, you can say in understanding you can say a proper molecular biological system of any species. Like you can use this protein mapping in any system. Okay, like in a plant also, if you want to know that how the growth is taking place, how the growth is affected by certain you can say environmental condition, then also you can do the protein mapping, extract the proteins, you can separate them, purify them, and you can quantify them, and then correlate that how with the environmental conditions, the protein concentration, the protein, but certain proteins are expressed more, or certain proteins are expressed less. So the protein mapping is just a special distribution of a protein, and is a very plays a very important role or key role in expressing the protein function. Okay. So let us start. Let us start with application in a medical sector. Okay, so this in a medical field, or we can say in in a medical field, these proteins normally act as a biomarkers. Okay, so if you have given or you have seen that there is a proper blood routine is done. Okay, so in blood routine, what does it is recommended? It is recommended to check the albumin and globulin level in a blood sample. Okay, similarly, if you look at a urine test, then also they suggest you to tested, the protein content is tested. Okay, so why this is done? You know, this are sometimes these albumin to globulin ratio, or you can say a protein in a urine, are indication of certain type of, you can say, symptoms of kidney disorder, or you can say a liver disorder. Similarly, liver functionality test is done. In the liver functionality test, what is done? Several enzymes are being tested. So what are these enzymes? These are all proteins. So these are all different types of biomarkers which are related with a certain diseases. Okay, they shows the symptoms, their you can say increase are associated with certain types of symptom of certain disease. Okay, apart from that, you've heard about that CRP. Have you heard about CRP protein? CRP protein. This is a C-reactive protein, okay, which increases when there's inflammation in your body. Okay, so this C-reactive protein is produced in liver, and when there's inflammation in the body, so this level rises. So normally, when a person is showing certain symptoms, so this protein analysis specific for this C-reactive protein is done. And if there is a more of C-reactive protein, that is the CRP is there, then it could be a sign of cancer or certain type of infections, or lupus, or arthritis, or tuberculosis. Okay, so if there is an inflammation in your body, then CRP protein level will be high. Okay, so this is just a simple example. There are a lot of biomarkers which are associated with the cancer also. Okay, so if certain protein biomarkers are uplifted in some of the cancer patients, then they are correlated with this specific type of disease. Okay. Now, similarly, protein analysis plays a very important role in the food sector. Okay, why? It helps them to enable the right. Sakshi, it's right. You have said that CRP protein is also related with the cardiovascular disease. If it is high, then there is a risk of various cardiovascular disease. Not only we can say a heart attack, but it is also related with other cardiovascular disease also. Okay. So I was talking about food sector. In the food sector, this protein analysis is related with nutritional labeling. So many of you have been buying the products, and you will be seeing at the back of the product, there is written about the quantity, written about the quantity of the protein. Have you seen it? Right. So because foods are made by combination of many of the components, so when the food is properly made after the processing, their protein analysis is done to enable them to help them for nutritional labeling. Apart from that, the pricing also get effects with the protein content. Okay, functional property is also gets affected with the type of protein you are using. Like sometimes sometimes a protein is required for, you can say, for swelling ability. Sometimes protein is required for your foaming properties. So all these functional properties are dependent upon the type of protein which you are choosing in your system. Now, next is the allergen labeling. Okay, allergen labeling is, many people are very allergic to certain type of proteins, like some are allergic to peanut protein, some are allergic to gluten. So in that case also, this labeling is required in the case of food sector when you are working with foods which are allergic to someone. Similarly, phenyl keto is urea is a disease in which people are recommended to take less amount of protein. In that case also, the protein labeling plays a very important role. For example, like gliadin and glutenin is very much required for the wheat flour for bread making. So in that case, if you are going for this bread making process, so you can optimize this conditions of gliadin and glutenin so that your bread making process can be achieved properly. Similarly, cheese products is highly dependent upon the casein. So the concentration of casein is very important when you are making any cheese product. So you have to focus on the casein content in the milk when you are going for the cheese product. So every time you are purchasing, every time in a company, when the milk is pulled out from the distributors, they are checked for this casein concentration. Okay. Similarly, egg albumin is also required for foaming. So these are certain proteins which are required, and every time the raw material is received by the company look after this protein content. Okay, in the initial raw material, then only they proceed for further processing. Okay. Now, this was about food sector. Let us talk about the genetic engineering field. You all know when we express anything in a certain cell, we need to check its expression level. Okay, and this is only possible when we know the proper analytical techniques. Okay, for example, there is a company called Medico which has transfected the leaves of tobacco with the SARS-CoV-2 spike glycoprotein, and they get a recombinant virus-like particle which self-assemble and burn off from the plant cell surface. Okay, so if this transfection has been taken from the day one to the day they are getting a maximum amount of this virus-like particle budding off from the plant cell, we have to analyze the protein. Okay, so from the day the plant has been transfected, we have to go day by day to check at what time this virus-like particles are budding off from the plant cell. Okay, apart from that, we have to check in which part of the tobacco this glycoprotein is expressed more. Okay, are you getting my point? That because plant cell has a very different, they have a leaves, they have a stem, they have a root, every part. So we have to check which part of your plant is expressing more of your protein. So this is only possible when you will be having analytical techniques. Okay, so all these things in the related to genetic engineering, this is highly dependent upon the analytical techniques, which I will be discussing in the later part. Okay, now in the pharma company, you all know it's all about the processing. This upstream processing means where the production is taking place, and the downstream processing is where the isolation and purification is taking place. Okay, so you have to look after, you have to check at each step that in the process of downstream processing, your targeted protein should not get degraded. Okay, so at each step, you have to optimize the process, you have to characterize, or you have to do the simple basic analytical protein analysis so that you get your targeted protein. Next is sometimes if you're designing any protein-based drugs. Okay, so in that case, characterization is very important. You know that if the drug during the processing, if the drug during the this protein-based drug during the process undergo aggregation means many proteins aggregate. Okay, so when they aggregate, what will happen? They will not be that much effective. Okay, so we have to characterize that they are not getting aggregated, they are not getting they are not getting modified. Sometimes certain modification takes place during the process, so we have to look after these things also. And next is the comparability, means if we want to compare certain protein with a certain protein based drugs, then also this protein analysis techniques are going to help us. Okay, for example, there is a drug, okay, a drug A, which is more effective against a, you can say, a cancer. Okay, and there is a drug B, which is also effective against a cancer. Okay, but it is least effective. Okay, bit least effective. So what you can do, you can compare the sequence of both the A and the B, and you can, from there you can define that what can be the probable reason why this B is less effective than that of A. Okay, so this comparability of how these proteins based drugs are working, that is also can be done by the help of the your can be done by your this protein analysis. Okay, some of you have talked about USP, DSP. See, when you express a certain protein-based, you can say protein, into any system, what is the main step? The first upstream step is the focuses on the production. Okay, so the maximum productivity. Upstream focuses on the maximum productivity. When you achieve a maximum productivity, then comes the downstream processing. Downstream processing, that you get a purified drug. Okay, purified protein-based drug. So what happens? The more the separation, the more you separate the protein from the impurity, the more are the chances that your yield will be less. Okay, so as your yield is getting less, you are taking something from a you can say standardized condition into a different condition. What happens? Sometimes your protein gets degraded in the process. It can happen, there can be certain proteases which are degrading them, or it can happen that due to certain environment changes in the conditions, like this buffer you are using, or you are the temperature at which you are doing the downstream processing is degrading your protein of interest. Okay, so how will you come to know that your protein of interest is getting degraded? It is only possible if you have a knowledge about the process, protein techniques. Okay, now characterization. Characterization, as I have told you, is dependent upon the protein structure and the protein function. So see, a drug, what does a drug do? Drug goes and bind to a certain receptor. Okay, so what does a drug do? Drug initiates a process, or it binds to certain receptors which initiate a certain process. So the proper structure of a protein-based drug is very important. Okay, so if there is any alteration in the structure of the protein-based drugs, it is going to affect the functionality of that product, functionality of that drug. So therefore, optimization at the purification, production and purification, once you have produced and purified, next comes the characterization. You have to characterize the structure of your protein-based drug. Okay, once you have characterized your protein-based drug, next comes the functionality. Now you will check that your protein-based drug is functional against a certain type of disease for which it is going to be used. If it is effective, what is its comparison with other drugs which are in the market, or other drugs which are there in the you can say in a company. So that comparability, that functional comparability is also can be done with the help of your protein analytical technique. And as I have told you, sometimes the presence of proteases, proteases are the enzymes which cleaves the peptide bond. Okay, so the name proteases is given to the enzymes which cleaves the protein, or the proteins, we can say the peptide bonds. Okay, so if the peptide bond will be cleaved, your protein will lose its property, not only in the terms of functional or biological, but also its structural integrity will be getting hampered. Okay, so the drug stability issue will be there. So you have to look after these proteases also, and this is also only possible with the help of your this protein analytical techniques. Okay. So till now, is it clear? We will move into the second part, that is the introduction to the protein analysis technique. Till now, it was clear to all of you. Okay, so now let us discuss with the second, that is the introduction to the protein analysis techniques. This is just an introduction. It is not possible to cover all the protein analysis techniques in such a short period. The course is there where the Learn to Upgrade will be offering you all the details about this protein analysis techniques. Okay. So the first step, as I was talking about, the first step in the protein analysis technique is your total crude protein content. Okay, so every time when you start working in a lab or in an industry, so when you take a sample, any sample, and you check the protein, that protein is called crude protein content because it is not a purified form. Okay, you have just taken a sample. This sample, as I have told you, it can be urine sample, it can be a blood sample, or it can be a microbial cell lysate. Okay, it can be anything. But the first step, the first analysis, the first determination of protein is known as total crude protein content. Okay, once you have done the total crude protein content, the next comes the particular protein. Like you have a protein of interest. Now you're focusing on that protein of interest. So every time you are going for isolation and purification, you are focusing on that particular protein of interest. Okay. So now you can be bit specific also, or you can go general also. If I say bit specific, means if it is an enzyme, you can use a substrate for that enzyme and you can go for analysis. But if you don't know anything about that protein, in that case, you have to go for a general process. Okay, general process I will be discussing, and all these analytical techniques which you are going to use is based on sensitivity, accuracy, the precision, the speed, how fast you are going to analyze those protein. And the most important aspect in the protein analysis technique is the cost of analysis. Okay, because I think when you do analysis, at every step you have to do the analysis. So your cost should be minimum, because if an industry is paying more cost in just in analysis of protein, it is not is feasible. Okay, so the cost of analysis is very important when we are working with when you're choosing any analytical technique. Okay, and the second is that speed. But in a very less time, if we can analyze more number of protein, then it is a best way. Similarly, the sensitivity means your accuracy and precision are very important. Means if you're analyzing, if you're compromising on the terms of the speed and cost, and your protein of interest is getting, you can say, is lost in the process of this analysis, in that case, that will be not a good way of analysis. Okay, so you have to maintain the sensitivity, accuracy, and precision. At the same time, you have to focus on less time and less cost of analysis. Okay, so there are two methods for total protein analysis and crude protein determination. So the first way is direct protein determination. This direct protein determination is done by, you can say, analysis of amino acid residue. In this, the your protein sample, crude protein sample, the extract you are taking is kept with the enzymes and for 24 hours incubation or 48 hours incubation. And once they have the digestion has been done, the amino acid, the chromatographic technique is done to separate this amino acid. And from there, the molecular mass is, the crude content, the protein content is calculated. Similarly, the indirect protein determination is the way, as I have told you, the elements, that is the element which we will be measuring in indirect protein determination is the nitrogen content. Okay, so, and sometimes what we do in indirect protein determination, we modify the functional groups of the proteins through a chemical reactions. I'm audible? I'm audible to all of you? Okay, so I was telling about direct protein determination. In direct protein determination, we keep this total crude protein and digest it with proteases. And after the digestion, we go for amino acid analysis. And from there, we calculate the protein content. And indirect protein determination, we focus on the focus on this nitrogen content. And sometimes we modify the functional group through a chemical reaction and that is the way of bioassays. Protein assays, which we do normally, is the indirect protein determination. Okay. So when we use this, measure this nitrogen content, we normally do by Zeldal method and Dumas method. Okay. So Zeldal method, this is measures the total organic. Okay. So in a crude protein, it can also measure, it will not only include just the protein, it will measure all the organic type of compounds, which N-containing compounds. So that's why in this case, the precision is not that good in the of protein content estimation. And the plus point is that it is very inexpensive. It is not that costlier. So it is normally used at the, you can say, initial scale to measure the protein content. And next is the Dumas method. Dumas method is that where they measure the total organic as well as the inorganic method. It is an expensive way of measuring the nitrogen content. Okay. Now comes the absorption of a radiation. You all know that there are certain protein assays, then UV absorption, you know, certain aromatic and amino acid have a property of absorption at 280 nanometer, and this helps in the estimation of protein content. So absorption at 280 nanometer is done. Normally tyrosine and tryptophan, you know, but sometimes what happens, the nucleic acid also absorbs at 280 nanometer. So in that case, this UV absorption at 280 nanometer will not give you an accurate results. Okay, so that's why the more the purified the sample, you can go for UV absorption at 280 nanometer. So if you remember, if you have worked with chromatographic techniques, you will be knowing that in the chromatographic technique, UV absorption at 280 nanometer is done because the more the purified sample, the more will be the accuracy. Okay. Next, sometimes protein assays. I know many of you have been worked with this protein assays. Can you name some of the protein assays? Many of you will work with this protein assay. Do you have an idea of this protein assays which are done normally to measure the total protein content? Pauline Laurie, right? Albumin assay. Albumin is not albumin is a protein. Albumin is taken as a standard. Alicia, no, Sadiq is not. Yeah, absorption is taken, but that is a different type of absorption. Biurate, right? Bradford, right? Okay. So apart from that, this protein assays, the other absorption technique like infrared spectroscopy are also be used, where the peptide bond, the presence of this peptide bond in protein molecule causes absorption of a radiation at specific wavelength, particularly in the mid or near infrared regions. So it is a very expensive equipment. So that's why in the initial stages, this use of infrared and NMR is avoided. Okay. Next is the scattering of radiation. You all know turbidity. The more the turbid the solution is, the more will be the scattering of radiation. From there also we can calculate the protein, determine the protein determination. Okay. So many of you have mentioned, so let us go quickly with this protein assays for the indirect protein estimation. There's a bicanoic acid or copper-based assay. You all know this is you can say peptide bond and specific amino acids bind in this type of cupric ion under alkaline conditions, and it has a very good sensitivity and widely used method for in the process of protein isolation and purification. But the problem associated with this copper-based assay is that this color is not stable with time. Okay, so as soon as you get start the process, you have to measure your absorbance. After that, you have Bradford or chromatographic blue protein assays. This in this, the basic and aromatic side chain are involved, get bind with this chromatographic blue G250 dye, and this dye complex absorbance is measured at 5995 nanometer. Okay. Now, apart from that, we have a Lowry protein assay. This is better in terms of others because the range I have mentioned, 10 to 1000 micro per ml of protein can be measured in the case of Lowry protein assay, but it is a bit time consuming. It requires a lot of time in comparison to other two. Other two can be done in a short period of time, and Lowry requires a two-step in comparison to other. In this, the protein peptide bonds with copper, and then we have to use a Falling Kelter reagent. After the incubation, we measure the you can say absorbance is at 750 nanometer. So this is a traditional protein assays which are normally done, which are preferred in because they are very less expensive and they are done in less time. Now, once you have analyzed the crude protein into your sample, next comes the method of protein separation. Okay, separation means you are isolating, you are fractionating now protein on the basis of their either size, charge. Okay, so the purity of protein normally improves as more separation steps, but as I have told you, in the downstream processing, as the more separation step or the purification step is there will be a lower recovery or lower yield. Okay, so the best method for protein separation depends upon the type of protein you are targeting. Okay, so the first initial step, the most commonly used step for the protein separation is precipitation. Okay, precipitation is a technique which is very simple and provide a very quick sample cleanup, especially in the lysates, cell lysates, or whole blood, plasma, or serum. And their operating principle is based on addition of organic solvents like ethanol, acetone, and then we can use a certain salts like ammonium sulfate, ammonium precipitation, right? And then we have isoelectric precipitation, means precipitating them at their isoelectric point when their charge is 0. So this precipitation is a very initial step which is normally done to clean up the, you can say, sample cleanup is done by this precipitation. After the precipitation, we can use any of these techniques like adsorption, size, adsorption based on the column chromatography, size. We can use ultrafiltration, we can use membranes of different molecular weight cutoff, and on the basis of that, we can use the ultrafiltration, or we can go for size exclusion chromatography, or on the basis of charge, we can use any of the separation technique like isoelectric focusing and ion exchange chromatography. Okay. So, because most of the time, the protein purification is based on chromatographic techniques. Okay, so let me tell you what is chromatography. Chromatography is a biophysical technique where the main prime aim is to identify, is to separate and purify the protein, and then use this purified protein for further qualitative or quantitative analysis. And in this chromatographic technique, we have a stationary phase. You all know this. There's a stationary phase. This is a column, okay, and the column have a stationary phase, and there is a mobile phase, okay. And this stationary phase is a solid phase, and mobile phase is a liquid phase in the case of protein, okay. So when there's a stationary phase, and when you load a sample, after the loading a sample, you add a mobile phase to it. So based on their interaction with the stationary phase, with the movement of mobile phase, your protein of interest can be separated. So the stronger the interaction between the stationary phase and your protein, that part of the protein will be eluted last, and the weaker the interaction between the stationary phase and the mobile phase, the protein will be eluted first, okay. But as I have told you, this column chromatography works on the principle of adsorption, means the interaction between the stationary phase and the protein will be based on the van der Waals force interaction. And in the case of charge, like ion exchange chromatography, the interaction will be based on their charge, okay. Like it can be an ionic ion exchanger, or it can be a cationic ion exchanger, okay. So the interaction will be based on the charges, and here the principle of binding will be on the adsorption forces. And when we work with the size exclusion chromatography, here we take a beads which have a capacity, or we can say a cutoff, molecular weight cutoff. So the protein which will be bigger than that of this cutoff will be eluted out first, and those which will be smaller will be remaining, okay. So this is the principle of chromatography. Now, after the protein separation and purification, so the next steps and the most crucial step is your protein identification and characterization. Lot of, you can say, although lot of biochemists have made significant advances in the protein analysis, but this is the most critical step. The purification and identification, or you can say characterization, remains a mammoth task, or you can say a formidable task, because it is the reason behind this, you can say, protein identification and characterization, is that due to the lack of a universal solution for macromolecule, that is a protein, because there's a lot of aggregation occurs, okay. Lot of aggregation occurs, and because of the charges and the structures, okay. One of you have told me to explain more about isoelectric. Sure, I will be explaining it. Just let me complete the whole slides, whole webinar. Then I will be explaining you at the last, okay. Just wait for a moment. So the protein identification, characterization can be done by using any of these techniques, that is two-dimensional gel electrophoresis or Western blotting, Edmond degradation mass spectroscopy, and we can use immunoassays like ELISA. This all can be done by this techniques, okay? Protein identification and characterization. So let me first explain about two-dimensional gel electrophoresis, okay? Two-dimensional gel electrophoresis is based on two technique. The first technique is isoelectric focusing, and the second technique is your SDS page. So this is a very powerful gel-based method commonly used to analyze complex samples, okay? And in this process, the first step, that is isoelectric focusing, is done. In the isoelectric focusing, what normally is done, there will be a gradient, pH gradient, and based on that pH gradient, and when there's electric field is applied, the protein get precipitated at their isoelectric point, okay? Let me show you one video, okay? Just wait to make you understand more, okay? Back in 2011, when I was happily doing my corporate job, I did two-dimensional gel. Is the screen visible to all of you? I'm audible? Is this? Okay. So this is a principle of 2D gel electrophoresis. As I have told you, 2D gel electrophoresis is, you can say, based on the idea, work on the two ideas, that is PI, that is isoelectric point, and another is the molecular weight, okay? So the first step in the 2D gel electrophoresis, what we do is a procedure for separating a mixture of proteins into spots on a gel. In the first dimension, proteins are separated in a tube gel. So we take a tube, okay? We take a tube, and then there's a gradient of pH is there, okay? So when we load a sample of protein in this, the protein are separated on the basis of their isoelectric point, okay? So this is a tube gel where we have a gradient, okay? And when we load a protein and we apply electric field with the cathode and anode, then on the basis of their charges, these proteins will be getting precipitated, okay, at their isoelectric point, okay? Is it clear? This is called isoelectric focusing, okay? In this method, different molecules are separated by their electric charge difference, okay? And this technique is a type of zone electrophoresis that is usually performed in a gel, okay? So is it clear? This is the first step, that is the isoelectric focusing is done in the case of 2D gel electrophoresis. May not contain a single type of protein. The isoelectric focusing tube gel is then laid on the top of a slab. So once you have done the isoelectric focusing, now you will place this tube gel, okay, on the SDS page. You know the SDS page, that is called the electrophoresis technique, where the separation takes place on the basis of molecular weight, okay? So in this part, the proteins, what happens? It can happen that 2 or 3 proteins will be having the same isoelectric point. It can happen, okay, that 2 or 3 proteins are having the same isoelectric point, but they will be having a different molecular weight, okay? So first, after the separation on the basis of isoelectric point, the second part is the separation on the basis of the molecular weight, okay? The proteins within the tube gel are electrophoresis into the slab gel and then move So you can see, if we take this part, the last end, this end of there are two proteins at the same isoelectric point, but they are having a different molecular weight. Is it clear? Like in this tube gel, this last one, there was a one band at the isoelectric point. Okay, there was a one band. But when we separated second time, there are two different proteins with the same isoelectric point, but different molecular weight. Are you getting my point? What is the difference? When we run SDS page, single SDS page, what we do? We get a separation based mostly on the molecular weight. Okay. When we are doing separately isoelectric focusing, that is on the basis of their PI, then we are getting basic on the isoelectric. But when we are combining both of these, first isoelectric combined with SDS, we can see that at the same isoelectric point, we are getting a different proteins with a different molecular weight. Okay. So this is the advantages which takes place in the case of 2D gel electrophoresis, and it helps in the proper identification of protein in comparison to other identification techniques for in the case of your other techniques. Okay, now let us in most cases, each type of protein forms a distinct spot that can be identified by staining them. So once you stain this, staining is a very important aspect in the case of electrophoresis. So once you stain, you will be getting a picture like this. The more this darker spots are the points where your protein has been. This darker point shows the protein. Okay, this black dot shows the protein. Okay, so normal Kumasi blue is used for staining. Okay, if the concentration is very less, then in that case silver stain dyeing is done. Okay, so after this, from here you can cut the sample. You can cut those protein part of the gel, and you go for further identification. Okay, so this is the advantages which holds with the 2D gel electrophoresis. So is it clear that how Western blotting is done? Sorry, two-dimensional gel electrophoresis is done. So now, after the when we work with protein identification, as I have told you, the identification based on intact alone, mass alone is difficult, because sometimes what happens that several proteins may possess a similar mass. Okay, several proteins can have a same mass. As I have told you that there can be a protein with a similar isoelectric point. Similarly, several protein may possess a similar molecular mass also. Okay, it can happen. So when the theoretical mass is calculated on the basis of their gene sequence, it rarely matches with that of the expressed protein. Okay, so for example, if you have expressed a gene sequence, on the basis of that gene sequence, you code down the amino acid sequence, and from that amino acid sequence, if you calculate a theoretical mass and you are getting a certain X amount of molecular mass, and it can happen that it doesn't matches with your mass of expressed protein. In that case, we have to go for other identification techniques like Edmond degradation and mass spectroscopy. Okay. Admin degradation is a technique, we can say is a technique where we use a reagents that are isothiocyanates, and it reacts towards N terminus of a protein. Okay, so in the admin degradation, we use a isothianate compound, and it reacts with the N terminus of a protein. And under suitable condition, the single round of admin degradation will cleave off the N-terminal amino acid residues. Okay, but the problem associated with admin degradation is that it is a very time-consuming method, and in this process of this admin degradation, we can normally have a limit. After certain limit of amino acid sequence, admin degradation cannot be performed. Apart from that, it can happen that if your N-terminal is modified, like if it is acetylated or glycosylated, in that time also admin degradation cannot be used, because it uses the free N-terminus for the amino acid sequence. So in this scenario of where admin degradation cannot be used, the mass spectroscopy opens up a new avenue. Okay, so this mass spectroscopy has replaced the cumbersome protein chemistry based protein purification techniques, and this mass spectroscopy works on the concept of peptide mass fingerprinting. Okay, this technique, mass spectroscopy, works on the peptide mass fingerprinting. Okay, so what normally is done in this mass spectroscopy, technique 1, I'm talking about the technique 1. Okay, in technique 1, as I have told you, we can take a part of our 2D gel, okay, and we can digest it with the specific enzymes, endoproteases. Normally, endoproteases are used, and then we digest this protein. In-gel digestion is done with a specific enzymes, and normally we use site-specific cleaving reagent that endoproduces like trypsin, and because of this sequence-specific proteolysis of the specific proteases, we will get a different type of peptides. Okay, and these peptides are detected by our mass spectrometry. Okay, and this you can say range of this you can say mass of different peptide masses are known as mass fingerprint. So you can see in this figure, this is a mass fingerprint. There is a different range written here, 980.47, 870.44. These are different ranges of masses which has been, fingerprint has been derived from this mass spectroscopy. So when we get the spectrum, or we get this fingerprint, peptide mass fingerprint, the next step is the protein database search. So once we get this PMF, we went to different protein database, and in this protein database, we try to match the fingerprint by using different database search engines. Normally, the two principal search engines are Mascot, Matrix Sciences, and Entity E, which is given by Water Corporation. Apart from that, there's a Sys, Prot, and Tremble are also there. These are different database search engines where we can search our peptide mass fingerprint with different fingerprint which has already been deposited in this databases. And from there, we can identify and characterize our protein. Now, apart from that, mass spectroscopy, the another important technology which is normally is used is technique 2, which is called mud pit. Okay, MUD, mud, PIT, pit. It is called multi-dimensional protein identification technology. Okay, in this technology of multi-dimensional protein identification technology, normally we combine it with liquid chromatography or capillary electrophoresis with the mass spectroscopy. Okay, so that's why it is called combination, because it's a multidimensional where we're having a chromatography section area which is attached to the MSMS, that is a mass spectroscopy. So that's why this is called multidimensional protein identification technology. And here what we do, we take a total cell lysate, that is cell lysate will be having we'll be digesting with the proteases. So once we will be digesting it with the proteases, we'll be getting a complex peptide mixture. Okay, so this peptide mixture will be separated on the chromatograph. Okay, by the chromatography technique. See, in this chromatographic technique, which I have given you, there are 3 phasic column. One is this reverse phase RP, reverse phase column. The first RP is a reverse phase column. Next is the strong cation exchange column. The second column is the strong cation exchange column. And after that, there is another reverse phase column. So see, there are 3 triphasic column is there. That's why it is called multi-dimensional. Means in a single process, you are using a different type of columns to separate your peptide mixture. And once the peptide mixture is separated, it passes through your mass spectroscopy. You will be getting a fingerprint, and from there you will be doing your cell analysis. Okay, protein analysis. Okay, so what is the advantages of such techniques? The advantages of such technique is that there is a large number of protein can be detected in a short span of time, because we are using a different type of column at in the same process. Apart from that, low abundance protein can be analyzed easily. And other is that the membrane proteins, which are normally not expressed in the case of 2D gel electrophoresis, can be directly being separated or characterized or identified with multidimensional protein identification technology. Okay, so in all these protein identification and characterization, the most major problem noticed during the analysis is the protein aggregation. Protein aggregation is numbers of protein get aggregated, okay, due to a certain type of interactions. Okay, so we have to look after, we have to measure this protein aggregate analysis is also important, and different types of techniques are used, like light obstruction, dynamic image particle analysis, microflow imaging analysis are done to check if there is a protein aggregation or not. Okay, if there is a protein aggregation, then there will be a different type of separating methods are used, like analytical ultracentrifugation, size exclusion chromatography, non-reduced capillary gel electrophoresis. So depending upon the aggregation, the process of your separation techniques will be totally different. Okay, so it is of prime importance that during the process you have to set a condition in such a way that protein aggregation should be minimum, because if the protein aggregation has taken place, your process is going to be costlier, and it will be going to take a lot of time. Okay, and it is going to hamper your protein stability also, protein of interest stability. So this was all about the introduction. Let us move to a small case study where protein analysis is playing a very important role in proteomics. Do you know what is proteomics? Proteomics. Right, study of protein. See, nowadays omics is a very common terminology. You will be knowing about genomics, transcriptomics, proteomics, metabolomics. Okay, so these are different types of omics has been studies has been done, and the main prime aim of all these studies is to understand the mechanism or the molecular aspects of any disease or any growth or development. Okay, right. So proteomics is the study of all expressed protein in an organism. Okay, all expressed protein in an organism. So proteomics will be fixed for an organism. Okay, so if I'm telling proteomics of a microbe, a certain bacteria, then it will be of proteomics, study of protein of that particular bacteria. Okay, right? So it will include all the presence or absence of specific proteins and expression levels over time, driven by certain external factors or disease state progression. Now, let us talk about Alzheimer's disease. Okay, I think everyone will be knowing of Alzheimer's disease. Do you know what is Alzheimer's disease? Alzheimer's disease is a very common cause of dementia. Okay, and where people everyday function gets deteriorated day by day, and it is morely related to the dementia. And normally 40 people across the globe have this Alzheimer's disease, and with the expansion in the lifespan, it is expected to increase. So there are two biomarkers related with this Alzheimer's disease, two protein biomarkers. Okay, so these two protein biomarkers are amyloid beta, okay, and there's a tau. So these are two biomarkers which has been studied, and you can say a lot of evidence has suggested that these proteins play a pathogenic role in the case of this particular Alzheimer's disease. So lot of therapeutic, you can say, development has been done based on these two biomarkers. Okay, so the pharmaceutical industries are mainly focusing on this amyloid beta, A beta, and tau. Okay, but apart from this biomarkers, the protein analysis has played a very important role in generating a global network, okay, where a proteomic approach is done to study the Alzheimer's disease brain. Okay, so what they have done in this paper, is by Raya Prolu. It is published in Neurophysiopharmacology in 2021. It is a recent paper. They have taken a 2,000 brain tissues. Okay, so they have taken a 2,000 brain tissues. They have done the genomic, epigenomic, and proteomic data. Okay, so we will focus on this proteomic data. We will not be going into the details of genomic, epigenomic, and RNA sequences. We will be focusing on the proteomics. So they are taking, they are using the local proteomics, means they are identifying the cell-specific protein analysis they are doing. That is how the protein is changing in the disease according to the cell. They're taking a neuron, they're taking a different type of cells of the brain, and from there they are studying the expression of the different level of the proteins. Okay, the protein changes which is occurring with the disease. Next, they are using a brain network analysis, using a control asymmetrical Alzheimer's and symmetrical Alzheimer's, and then they are using the biofluid proteomics, means they are using your plasma, taking the plasma also, that is your blood samples also, and then they are using a cerebral spinal fluid also for studying. And then they are using this three of them to define the protein biomarker for this particular AD brain. Two have already been used, that is your amyloid beta and tau. Apart from that, they are using this concept of protein biomarker by analyzing the protein. Okay, and from there, they are measuring the changes which are having in the disease, by measuring the inflammatory biomarkers, synaptic biomarkers, vascular biomarkers, myelination biomarkers, and the mitochondrial biomarkers. So from here you can see that protein analysis can help you in choosing the biomarkers for a particular type of disease. This is just an example. This is just a conceptual framework for translating brain protein network into a clinical biomarkers. So this framework which is given by this paper is to make you understand that by choosing the proper protein and by analyzing a whole sum of protein which is expressed by any system, like it can be your CS, the protein in the CSF, protein in the plasma, protein in the cells. They can give you a understanding of about the biomarkers which can be used for the clinical studies. And how they have done this, they have used two analytical techniques, that is mass spectroscopy for this, that is mass spectrometry. You know, it is antibody free, it is very highly sensitive and accurate, and relative and absolute quantifications can be done. And immunoassays, it is a bit costlier, because for that you have to have antibodies for those proteins for which you are studying. Aptamers are the nucleic acids which bind to the certain proteins, so you have to design those nucleic acids. High sensitivity and accuracy is there, and moderately selective target detection by enzyme-linked antibodies. So these are two of the characterization or identification steps that can be used to generate the clinical biomarkers for the Alzheimer's disease brain. Okay, is it clear what I'm trying to tell from this conceptual framework? That this is just a framework for Alzheimer's disease brain, but you can use this conceptual framework for other type of disease also. Okay. Okay, thank you for your responses. Okay, with this, we have completed the second part. Now let us come to the section of scope and opportunities related with this course. See, this course is going to benefit in all your competitive exam. You all know that the most questions related in the competitive exams are related to your proteomics, your translation, your protein analysis. So this course is going to benefit in this your competitive exams, you know, CSIR, NET, GRF, DBT, GRF, ICMR, GRF, GATE. So this will help you if you are going for this course or you are interested in this protein analysis. And if you want to work in this area of protein analysis, there are different job roles which are offered at the industry, like protein analytical officers, research analyst, data analyst, operation analyst, bioanalysis scientist, process system analyst. And these are the of jobs which you can get in the different industries, like in the food also, in the pharmaceutical company also, even the companies like BioRed, Water Corporation, Thermo Fisher, which design this type of instruments, like who design the columns, who design your sensors. All these roles or job roles can be offered if you have a proper understanding of a protein analysis and data interpretation. And this course is going to help you in this domain. Okay, with this, I have come to the end of the session. Okay, hope you got benefited what I covered today. So I have shared you the link for your review. Kindly give your review if you like this presentation or webinar. Niranjan, the presentation will be with at the Learn to Upgrade. From there you can see the recordings. Recording will be there. Okay. Thank you, Aditya.